Okay, so there are two types of reproduction, asexual reproduction and sexual reproduction. Asexual reproduction only involves one parent. And so mitosis, which is what we're going to learn in this video, is considered asexual reproduction. Where sexual reproduction involves two parents, and that is going to be meiosis, and that will be in the third video. So the question is, how do little elephants grow up to be big elephants? Another question we could answer is, why do animals shed their skin? And the answer is by mitosis, or asexual reproduction. And the process of asexual reproduction begins right after the sperm fertilizes the egg. Here are three reasons of why cells will reproduce by asexual reproduction. One is for them to grow. One is to repair cuts or scrapes or anything that's damaged. And the third one is to replace dead cells. Skin cancer is when asexual reproduction or mitosis is going out of control. And skin cancer is the #1 cancer that kills Americans. If you go to cellsalive.com mitosis, you can see an animation of the mitosis or the cell cycle. We will be going through each of these phases: interphase, prophase, metaphase, anaphase, telophase, and then cytokinesis. Okay, so the first phase we're going to talk about is called interphase. And interphase is actually not a part of mitosis. It actually occurs before mitosis begins. There are three parts of interphase, G1, S, and G2. And so this is when the cell is growing. The DNA is being copied. That's what S stands for. S is synthesis, which means something being made. And then after the DNA is copied, which takes a long time, then you have G2, which is when the cell prepares for mitosis. So on your little oval there, you need to draw in chromatin, because that's what you would see during interphase. You need to label the nucleus, which is where the chromatin would be located at. You would, just for reference, to see where the cytoplasm is, label cytoplasm. And then the outside of your oval, that is your cell membrane. So during interphase, you can see the nuclear membrane, which I need to draw in, and the nucleolus is also visible. So please draw both of those in. So the nucleolus is right there, and the nuclear membrane is right there. So please make sure your diagrams are nice and labeled. Here's a picture of what they would look like under the microscope. Interphase, animal cell and plant cell. It's a little bit difficult to see, but you should be able to see the nucleolus, the darkened area in the cell, and you should be able to see the nice circle of the nuclear membrane. Okay, so now mitosis is going to begin. And so our first phase or step of mitosis is called prophase. Cells are now starting to divide. And so now, instead of being chromatin visible, we have chromosomes visible. So our chromosomes are right here. We can also label that as sister chromatids, remember. Also visible are centrioles. So our centrioles are kind of forming, you can see the pieces of them right here. There's 4 of them. 4 little pieces of them in their picture. And then you also can see spindle fibers. And spindle fibers are going to start forming between the two sides of the cell. So please label your diagram. And then up here I just made sure you realize that the nuclear membrane and the nucleolus are disappearing. They are no longer going to be visible by the end of this phase. So do not draw them in. Here's a picture of what it would look like under the microscope. So an animal cell, you can see the little lines of the chromosomes. The nuclear membrane isn't quite as distinct as it was in interphase. And then you can see your plant cell as well. All right, second step of mitosis is metaphase. And in metaphase, it's very, very clear because the chromosomes are all aligned in the center of the cell at the equator. They're all attached to the centromere. So you got your spindle fibers coming from the centrioles, going through the centromere, and then to the other side of the cell. All these nice chromosomes are all in a nice line. So if this was a human cell, you would see 46 of those chromosomes all in one long line. Under the microscope, sometimes I might just like a big smudge of chromosomes there, but you can see the nice spindle fibers, right here. And in a plant cell, it almost looks like a baseball stitching. Okay, the third step in mitosis is called anaphase. And so in anaphase, our chromosomes split in half. And so they are now chromatids. The chromatids move, being pulled by the spindle fibers to the poles near the centrioles. So in your picture, please draw your centrioles on each end. Your spindle fibers are shorter and then you have the chromatids attached to them. Here's a picture of what it would look like under the microscope. They almost look like pom-poms or little spider legs on each side for a plant and an animal cell. Okay, the last step of mitosis is telophase. Telophase is basically the opposite of prophase. It is the fourth step in mitosis. This is when your nuclei are actually going to form or reform. So now our chromatids, or our chromosomes, sister chromosomes, are chromatids are going to appear as chromatin. So by the end of telophase, they're going to be back into the thread-like structure of the DNA. And mitosis is going to end by the end of telophase. So in your picture, you have your nuclei, you have your chromatin, but also realize that your nuclear membrane is going to reform around the DNA, and the nucleolus is going to reform by the end of telophase. Also, the spindle fibers will be gone, and you will no longer be able to see the centrioles. One thing is that telophase overlaps with cytokinesis, so let's learn about that next. When looking under the microscope for telophase, you would see two distinct nuclei. Sometimes in a really good microscope, you'll see the stretching of the cell. Okay, in order to divide the cells into two, we have to go through cytokinesis as well. Cytokinesis is the division of the cytoplasm, and it overlaps with telophase. So it happens at the same time and near the end of telophase. Basically, the cell membrane needs to move inward to basically create our two new cells. These two new cells are going to be called daughter cells, and they're each going to have their own nucleus at the end, and they're going to have identical chromosomes due to the copying of the DNA in interphase. The way each cell divides its cytoplasm is different. In an animal cell, it's going to form a cleavage furrow, and in a plant cell, it's going to form a cell plate, which will then become the new cell wall. So this top picture is the animal cell, and you can see this is telophase. And in order to divide the cell into two, we're going to form this cleavage furrow. So the cell membrane actually pinches in, and eventually will snap, and then we'll have two new cells. In a plant cell, however, it doesn't work like that. We need to form a new cell wall. So we're going to put cellulose deposits here, and then eventually that will seal up, and we'll have a brand new cell wall. That when the cellulose is building, that is called a cell plate. So next in your outline, you have just an overall view of animal mitosis under the microscope, and one for plants. You need to remember the order of these phases. It's Ipmatc. I pray more at the church. I pretty much am totally clueless. But remember, these letters stand for the whole cell cycle, not just mitosis. Just mitosis is PMAT with the C. Interphase is not mitosis. So here's a picture of the cell cycle. It starts with interphase, which is not mitosis, and then you have mitosis in blue, where you have prophase, metaphase, anaphase, telophase, cytokinesis, and then that cell will start going through interphase and start going through G1 again and start growing. Here's another diagram. Here's your interphase, G1, S, and G2. This is all interphase of growth, DNA replication, and more growth and preparation for mitosis. The PMAT part is just a tiny part of the cell life. PMAT, mitosis, and then don't forget you do have a cytokinesis right here. So please, after this video, go to cellsalive.com/mitosis and watch the mitosis animation. Okay, in a eukaryote cell, we can control our cell division by protein. There are actually 3 checkpoints in a cell to double check to make sure that things are going correctly. There is a G1 checkpoint. So when the cell is growing, the proteins in there will check to see whether the cell is growing good, it's healthy, and is large enough to divide. If it is not, it will stop it and wait until it is ready to go. So then the DNA will copy, and then after G2, there's another checkpoint where proteins check to make sure that the DNA is copied correctly so that it can go through mitosis properly. And then at the end of mitosis, if the cell divided correctly, then there are signals, this is all done by signals, that were made to exit mitosis. When control is lost, results in cancer. Cancer is the uncontrolled growth of cells. It may result if cells do not respond to these control steps, these mechanisms. It can also be caused by mutations in the DNA. And this concludes video 2 on mitosis.